Okay, so then let's start again with neurocognitive methods. And as you realize, there was this audio artifact on the old video, so as I said, I deleted it, and we start afresh, which is also fine. Yeah, and let's just go on and talk about what we have seen and what will happen. So the first thing that also was missing last time is this is the book that I mean. Please read it. Yeah, so that's kind of the task for the winter term in this course. Read this book to follow the class. And everything that is on the slides is, or by and large, from this book. We discussed then last time that there are different neuroimaging methods that can be looked at in terms of their temporal and spatial resolution. And we discussed some of these in a little bit more detail. And obviously, we are interested in fMRI for this course. Again, the course outline, basically covering everything and nothing in a way. So we basically cover everything from signal generation to data analysis, but of course only to a certain level of depth. So this is what we want to do before Christmas. So you will have heard many things, but your knowledge will not be enough to really start working on anything. But at least you have heard a lot of things and kind of have charted the territory of what you want to study in more detail. And then the idea of this first section was, so this introduction and overview to go over everything already from signal acquisition, biology, and data analysis in even a shorter unit, so that you know also what the whole course will be about. And we started with that last time, and we discussed the physical foundations of magnetic resonance imaging to the shallowest level possible. So we discussed that this term magnetic resonance imaging comes about because there's a strong magnet involved, this is created by this electromagnet, which needs helium. So we talked about this. Then this magnetic resonance imaging, this resonance term comes about because there's something that is often referred to as radio frequency excitation of nuclei, and then there's some resonance and then emission of energy by the nuclei at the resonance frequency, and this is where this resonance term comes from, although I think it's not really understandable what this should mean at this level of explanation. But at least this excitation reception and doing at a resonance frequency is where the resonance comes from. And we talked about that this can be done with these different coils, where these phase array coils are the most popular these days. And then we talked about that the imaging comes in because what you actually can do is also manipulate the magnetic field over space, so get kind of a spatial encoding by means of varying magnetic fields. So this is where the terms come from. The MR setup. Has anyone been to the CCNB lab last week? No? You have been. How was it?
Well, like any MRI scanner, I guess. They all look the same.
Well, yeah, that's what I once told a famous professor when I didn't go to our MR scanner with this other famous professor, and it was a big issue. Anyway, so one should always show the guest the MR scanner. Anyway, so the more serious thing is that these MR scanners, are kind of very powerful and very strong machines. So they have this strong magnet, which can really have harmful consequences if one is not careful and walks in there with metal that is drawn to the scanner. There is, if you have subscribed to the CCNB email list by now, there is a safety seminar on November 13th or so. I think it's a Tuesday at 3 p.m. I know that you have Adrian's class at this time. So you might want to discuss with Adrian that you attend this safety seminar, because it's actually it's kind of part of the schedule. And if you want to, within 1 year, do something on MRI, you should have attended this safety seminar. Who has gotten this email? The Christian Kainz. So everyone else, please subscribe to the CCNB email list, so that you also get the emails that go in this direction. So MR safety is an important concern. The magnet is never switched off, so don't just work in there because it can really have harmful consequences, and this needs to be avoided at all costs. So this is, I think, how far we got last time. And of course, this is just a very shallow introduction to anything. But then at least you know that there's something like this MR scanner, and it involves magnetic fields. The next thing then, where we also want for now a shallow introduction, is actually what is measured and how does that relate to neural activity. So when you read fMRI papers and hear people talking about fMRI, they will often use the term BOLD signal. BOLD being the abbreviation for blood oxygen level dependent whatever. So BOLD signal or BOLD contrast are sometimes used, which where the important thing is that what these images that are acquired and these data analysis reflect are changes in the blood oxygen level and not neural activity per se. So it's not action potentials or excitatory postsynaptic or inhibitory potentials, but it's a reflection of the metabolic demands of active neurons. Yeah? So it's an indirect measure of neural activity. Always has been, and BOLD always will be. The reason why one can measure that is because, of course, the brain needs energy and is one of the organs that doesn't store energy, so it must be continuously be provided. And of course, energy is created in living systems, not all living systems, but at least in mammals, by for example, the combination of oxygen and glucose, and we will look into that in more detail from a biological perspective. Oxygen and glucose can be used to create ATP, and the oxygen is delivered in this case via hemoglobin. The neurons need the oxygen, and the hemoglobin, if it has oxygen bound to it, it does not have a magnetic moment, so it's diamagnetic, so it will not affect a magnetic field. But if it's deoxygenated, so if it has released its oxygen to cells that need oxygen, then it becomes paramagnetic and leads to distortions of the magnetic field. And these distortions or non-distortions by oxygenated or deoxygenated hemoglobin of the magnetic field, this is something that can be detected by means of MRI. And here it already uses a term that we haven't talked about, so it's actually a decay of the transverse magnetization that depends on the local hemoglobin oxygenation. So this is just, so there you will find that in these slides, there are some slides that have a lot of text on them. There are other slides that have pictures on them. The text slides are not so much that I have never heard that people say you should not put too much text on PowerPoint slides. These text slides are there for you to have something that you can memorize in a condensed form. So these are, I think, they are essentially there to help you, or basically a study cards to memorize things, whereas the slides that have pictures on them are more for you to appreciate and think they are beautiful. I don't know. So the bold signal, and basically using MR for this indirect measuring of neural activity, is around for by now almost 30 years. So the first experiments in humans using this technique were done in the early 90s. MR, of course, had been around for longer, so that has been around. So MR imaging in a clinical context since the 80s, early 80s, essentially, and also the idea that neural activity and changes in blood flow, essentially in the brain, occur. Had also been around for longer, so it's not that in the early 90s, at the same time the MR imaging was invented, and it was found that actually there's a connection between neural activity and blood flow. That was not the case, but at this point it's kind of because scanners became available to humans, and because there were some new developments in terms of sequences for MR scanning. It's that these early fMRI experiments were made. What this shows is a block design, as one would call it these days, where humans were stimulated with bright visual stimulus, and then sometimes it was off and sometimes it was on, and this shows measurements from the occipital cortices. And what you can see here is that the MR signal goes up if the stimulus is on, and the same thing is shown here. This was then also done in a different domain where people don't use their fingers, and here where people move their fingers, so do a motor response. And if one looks at the signal recorded from motor cortex, one also sees that the signal goes up. And then there's another one that is again a visual experiment that shows that again under visual stimulation the MR signal increases in visual cortices. Yeah? So these are the early pioneering human fMRI studies. Essentially, these experiments, they don't explain the whole thing, how neural activity is induced by visual stimulus, how this neural activation is somehow related to vascular changes, how this changes blood flow, how this then changes oxygenated hemoglobin and so on, and how this affects the MR signal. This is not what these experiments explain. What these experiments explain is, or show, is that if you turn on the light to a human subject, then the MR signal using certain sequences in the visual cortex goes up. Yeah, and this has been reproduced many times. I have reproduced it myself actually, but everyone who has ever done an fMRI study reproduced that, and this is something that really, really works. So sometimes, of course, you have people questioning the whole validity of fMRI, and that's of course fine. One should, of course, question everything and be critical of everything. But the fact that visual stimulation of human participants increases the MR signal in occipital areas where visual areas of the brain are, I think this is, at least this, I don't know anything else about neuroimaging, but this is actually an effect that exists, and this is definitely real and has been reproduced many times since then. So this tries to explain at this point already a little bit more in detail of how one thinks that these kind of changes play a role. So we will go about this again. So for the moment, the important thing is that local brain activity leads to local changes in oxygen consumption and cerebral blood flow, which again changes the ratio of oxyhemoglobin and deoxyhemoglobin. One thing you might also get used to at this point is that neural activity actually leads to an increase in oxyhemoglobin and a decrease in deoxyhemoglobin, which is a little bit surprising because you would think neural activity eats up all the energy, but what actually happens is that there's an overcompensation, and where there's demand for energy, there is an increase of the oxyhemoglobin. And at the same time, a decrease of the deoxyhemoglobin, which then leads to the fact that the paramagnetic, so the hemoglobin that influences the magnetic field goes down. So there's less distortion, and the MR signal goes up. So and then you have the nice thing that brain activity goes up and MR signal goes up, although in between actually what happens is the one that interacts with the so the deoxyhemoglobin that interacts with the magnetic field goes down. We will get to that again. It's one of these things where one thing goes up, the other thing goes down, and then another thing goes down, and then the whole thing goes up. So it's nice for exams, but otherwise it's not particularly demanding. So now the question is, this are kind of very much the physical and biological foundations, and I don't know whether you have started to read papers in fMRI. Who has read an fMRI paper by now? Good. Who in which of this paper used a GLM analysis? Okay, so what kind of analysis was used? No analysis?
I think we didn't get the question. I think everybody would have put their hand up.
Or maybe we just didn't really agree with the results.
So were there blobs on the brain in this paper? Yes, maybe. Who knows? Well, the question is, so what I'm wondering, so what I'm trying to say is that the important thing, or what most fMRI papers actually focus on, is not so much these physical and biological foundations. So they might not talk about that the bone signal is an indirect measure of neural activity and that it has to do with blood flow or so, something like that. It's likely that a typical neuroimaging paper will not talk about this. Typical neuroimaging paper will also not talk about I mean, there will be a sentence of how the data was acquired, but it will not go about like that you need the Fourier transform to actually do MR imaging or something. This is not what you will find in a fMRI paper, but what you will find in a fMRI paper is how the authors analyzed the fMRI data, right? So was there a description of the analysis of the data in your papers? So can again these people who read an fMRI paper raise their hand? So who remembers the analysis? So how was that analyzed in your case?
It depends on which paper, but if I pick one, maybe GLM.
GLM, good, thanks. Done. Yeah, so definitely it depends on which paper. That's also an important point, because not all fMRI papers analyze the data the same way. But one way to analyze, and a very popular and a very basic way to analyze the data, is using the general linear model, as we will then also discuss in January mainly, but also a little bit before Christmas. So what I'm trying to get at is that in functional neuroimaging, cognitive neuroscience, cognitive neuroimaging, a lot is about data analysis, and actually a lot more than about biological foundations or physical foundations. There is research in the physical foundations, of course, there's research in the biological foundations, but the cognitive neuroscience community, or the brain imaging community, or the brain mapping community, whatever you want to call them, they focus, and including myself, they focus on the data analysis part. So, and this is what this last thing is supposed to be introducing here a little bit, by first talking about what is actually the data that we, as cognitive neuroimaging people, researchers, computational cognitive neuroscience people, of course, what is the data that we are dealing with? So there something important is that you basically learn that from an MRI scan you can get different terms of images. There's one form of image which is called a structural or T1 weighted image, and what this T1 means we will come to in due course, and this is shown here. So this is typical T1 image. It's a beautiful anatomical image, so you can see gray and white matter nicely. You can see the basal ganglia nicely. It has quite high resolution, usually like 1 millimeter spatial resolution. It's great, but this is not the stuff that is actually used for fMRI. So for fMRI, how do I get back this? For fMRI, what is actually used are so-called T2 star weighted images or echoplanar imaging images, which have lower spatial resolution, usually commonly, let's say 2 by 2 by 3 millimeter voxels. So voxels just being the equivalent to pixels. And of course here you just see a 2D image. So essentially here you only have pixels, but you can imagine that this is a 3D volume because you have many more slices. Now the thing with functional MRI is that here from an anatomical scan you get one image, which is a nice anatomical picture of your brain. From a functional EPI scan, you get a time series of images. Yeah, so you acquire each of these functional images in over 1 or 2 seconds. Yeah, so this is so you can make a picture of the whole brain at this resolution every, let's say, 2 seconds. And then if you do that, you get a sequence of images, 3D images that reflects for each location in the image a time series. Yeah, so that's fairly important that you appreciate that functional imaging data reflects a time series. And then what you actually, in terms of the images that you always see in papers, what you see there then are what is often referred to as statistical parametric maps or SPMs, which are usually T statistics, so T values, which result from the analysis of this time series and are then often actually not resulting from only one participant but from multiple participants. So if you have a group study that you're looking at, and then what you see in terms of these blobs, so that's nice. In terms of these blobs that you see overlaid here, are just some surviving high T values that result from usually GLM analysis of these data overlaid on a single anatomical image. Yeah? So basically, when you see these images, what has happened is that a time series of data, often from multiple participants, has been statistically processed and evaluated and is then compressed and represented in this kind of form. There are different ways to show that. So another way that's also often used is to put them on top of an average anatomical image, which is a little bit more maybe reflecting the spatial uncertainty that you have here, because clearly this is a little bit overconfident in terms of the spatial location, because these functional blobs here, they were not acquired at the same resolution as the anatomical data usually. A little bit even more fancy, if you do more image processing, you can do 3D reconstructions from these kind of images. So basically taking these 2D images and putting them through an algorithm to create a 3D mesh, and then you can also overlay these kind of activations onto a 3D mesh. Yeah? But it's important that you kind of, on first approximation, distinguish between the results of analysis, the data that actually gives rise to these analysis, which is the EPI time series, and the anatomical images. Yeah? questions about this right now? Who knew that all along? No, be honest. I mean, who knew that? Okay, good. Then I kind of know what kind of what's going on. Good. So to get a little bit more of a feeling of how the data is arranged in fMRI, this helps quite a lot. So in general, how things are done in fMRI studies is that there's a cohort of participants, or sometimes called subjects, and participants come to a scanner and participate in an experiment like you maybe did last week. And I encourage again everyone to participate in the studies that are ongoing. So they lie down in the scanner and get exposed to a certain cognitive task. So what did you do?
It was a visual task, so it was scrambled pictures, and I simply just had to watch them, and then there was like a tiny task where I had to press a button when like the fixation cross changed color.
So with whom was it? Daniel?
Yeah.
Yeah. So what's your name? Sorry. Zina. So Zina saw a series of pictures, and some of them were scrambled, and some of them were normal pictures of scenes, maybe skyscrapers and woods or something like that. I don't know. But that was over time. So she lay in the scanner. These visual stimuli were presented, and at the same time, EPI data was acquired. So you heard this beep, 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 which reflects the scanner noise, and actually one of these beeps is always the acquisition of a volume. And this, while you were lying in the scanner, you maybe did a couple of runs. So how many runs did you do?
I think 7.
7. That's and they were like 5 minutes, 6 minutes.
10.
That's a typical.
10 minutes.
10 minutes. Wow. And you did 7?
Yeah, it was long.
Wow, that's typical. That's kind of why I did that as well when I was younger and when I was working in vision. So in vision, in general, you always want a lot of data, but in vision, especially, you want a lot of runs. And runs means basically a continuous time of scanning. So scanners turn on. It does its data acquisition. The participant does the task, for example, in this case, looking at the picture and pressing the button when the fixation cross changes for 10 minutes. And then there's a break. But Zina had to stay in the scanner, could relax a little bit, and then after a little bit of a relax time in this very spatially confined thing, she decided, okay, let's go on, and then he did the next run. So in each run, so this is somehow here representing different runs. In each run, what happened was that actually, like I said on the previous slide, a series of 3D images was acquired. So this little thing here is supposed to be a, oops. I want to also use my pointer while zooming in, but I think that's not allowed. So the I want to try it again. Can I now also zoom in? No. So during a single run, a series of 3D images was acquired every 2 seconds or 1 second. 1.5 seconds or so, one 3D volume was acquired, and this is what we call a volume. It's also called an image. It's also called an EPI image, EPI file. So if you analyze the data, that's typically the file resolution that you get, and it basically reflects the MR signal of all locations in the brain that you image. These volumes are typically acquired not as really 3D volumes, but as a series of slices. So this is where these slices come from. So the 3D object here, the brain, is represented by, let's say, 30 or so 2D images, which are these slices. And each slice then, of course, refers to a certain location in the Z direction, so in the up-down direction. And each of these slices then is an image and has, as every image, pixels. And because these pixels have also a 3D kind of feeling to them, because they are from these slices, they're called voxels. Yeah? So that's the hierarchy of how data is acquired. And this is quite important, so you should really get this into your system, otherwise you have problems understanding fMRI data analysis. So you really need to appreciate that for each location in the brain, there's an image made, and this image is actually called voxel, and for each voxel you get many time point measures. And this is also shown more here. So this shows this in some different representations. So you have these voxels. Now here I plot a simulated time series over voxels. So you, of course, you have many locations in such a slice, but because you acquire the data over time, for each single location, you get many time point measures. Yeah? So then you have a signal that has both spatial information, because you know which voxel you acquired that from, and temporal information, because this was acquired over time. Now, of course, the temporal information is quite important with respect to the task, because at some point Sina saw pictures that were not scrambled. So she saw X, and sometimes she saw then pictures that were scrambled, so scrambled eggs. So then, of course, the researcher is interested in whether there are some differences between these scrambled images and the normal images in terms of the neural activity they induce. And of course, this then needs to be coded in terms of the time at when they were acquired. Yeah? But so this is really important that you appreciate that you get what I also call MR signal time series, or voxel time courses. So voxel time course is actually a Brain Voyager word, but I think it's helpful. So for each voxel, each 3D location that you image, you get a time course. Now, based on this, one can do different kinds of analysis, and the important thing is that you appreciate that all of the stuff that you find in papers, whether they show you these kind of blobs on the brain, whether they show you time courses extracted from certain regions of interest, whether they show you classification accuracy for or decoding brain reading for the task that the participants did, or whether they kind of do what we will deal with in the summer term. So they do a real time series analysis and fit differential equation models to them and inspect their parameters. Everything starts from this kind of data. Yeah? So everything starts from voxel locations that represent space, and for each voxel having a MR signal that evolves over time, and of course knowing what the participant did at a given moment in time. So when these images were presented, of course there was a computer that presented these images, and this also time logged when these images were presented with respect to when the fMRI data was acquired. Yeah? So typically for a given run, one sets the clock to 0 when the run starts, and then a certain image or a certain task is done, or a certain stimulus is shown at, let's say, 20 seconds into this run, or then 150 seconds into this run. So that one always records that. And then here, what I also have on this slide are different forms of analysis. So this is kind of the most old school way of analyzing fMRI data, which is very much inspired also by EEG analysis. So maybe you know that in EEG analysis, you basically cut out, for every stimulus, you cut out the peristimulus signal. And of course, you can do the same with fMRI data, and then cut out for a given stimulus group. Here there are locations, faces, colors, and objects. You can cut out the signal for all the stimulus presentations, average them, and then average also the data from a given brain area, and then look at these evoked responses. Yeah. So this is now the average over multiple presentations of locations, faces, colors, or objects. And you see that for this area, for example, locations yield the strongest response. So this is 4 to 6 seconds after the stimulus was shown, there's a strong response for locations in this area. For this area, there's a strong response for faces. Yeah? So this is kind of what is called the event-related and ROI-based approach, which was popular kind of mid-90s to late 90s. This was also popular together with some adaptation experiments where you show some pictures and then you show some other pictures which are kind of similar but not quite, and you look whether there's a release from adaptation. These were not on vogue for quite some time, but then like 2 years ago I saw Tim Behrens doing adaptation experiments again. So it's like neural networks. They were on vogue in the 80s, then everybody hated them in the 90s and 2000s, and then in the 2010s they are back again, are called deep learning, and neural networks are hip again. So things go in and out of fashion. The more principled approach, but essentially the same thing as that, is the GLM approach. So here, instead of cutting out stuff, one does the cutting out using a model formulation approach, basically a design matrix, and using parameter estimation, which essentially does the cutting out. And then also projecting these kind of differences that you see here in the peristimulus time thing to actually measure them against the measurement error, and then plot these differences as t-statistics on the brain. Essentially, the GLM analysis looks at the same kind of differences as the event-related averaging approach. A little bit different in terms of the philosophy is that you don't look into regions of interest, but you look into the whole data set, because generally you do whole brain imaging, so you measure the whole brain, so it makes sense to also look at the signal from the whole brain. These days, this of course still happens. So you see these pictures often also in our own work. They are now supported by cognitive computational modeling, where you actually develop a mathematical model of the task that the participants are doing that evolves over time, and then you correlate aspects of this with the fMRI data. This is what I've shown you in the very beginning about this work that we did with the agents. This is what people call functional, what people call computational fMRI or computational EEG also, but in the end it's a GLM analysis. It's called masked univariate because you do the same analysis for each location in the brain. It's sometimes done in a Bayesian way, but hardly. It's mainly done in a classical frequentist statistics way. It's sometimes supported by what is called small volume correction if you want your p-values to be more significant. And it's sometimes also done as PPI, which is also a GLM, but speaks more to connectivity approaches. So who has heard the term PPI? You all heard the term PPI, because last week in the talk, Philipp Sterzer gave he showed you some PPI data. And here actually, I think they also used a cognitive computation modeling approach. Yeah, they did. They used this awesome hierarchical Gaussian filter, which he did not explain in detail, but it's awesome. And yeah, so these are two approaches, which, and yeah, maybe also saying about this. So the GLM approach was developed in the mid-90s, is very closely associated with the work at the FIL, the Functional Imaging Laboratory in London in the mid-90s, even more so associated with Carl Friston. It's not that people did not analyze fMRI data using the GLM before it was implemented in SPM. Of course they did. The GLM, and we will get to that, or you maybe have already started watching the lectures from last year, is essentially just a unifying perspective on classical statistics. So you can also think about this as doing t-statistics or t-tests at every location in the brain. It's essentially the same thing, but it's formulated in a different way, and it's formulated in a more flexible way. So that's kind of mid-90s, basically up to this day. Still popular, the Bayesian stuff never really caught on. PPIs, always popular. This is kind of more the novel thing. Yeah, can debate how novel that is. And then in the mid-2000s, actually already earlier, so in 2003, and also I think there's also stuff from this in the 90s. I think there's the Edelman stuff on multidimensional scaling from the 90s, but really from the mid-2000s on, because there were two papers in Nature Neuroscience, one by the Kamitani and Tong people from I think mainly MIT, and one from John Dylan Haynes and Garen Rees from London at that point. They figured that you can not only do GLM, but you can also look at the pattern and try to classify the pattern using a classification approach, for which you can use all sorts of classifiers, for example, Fisher's linear discriminant. You can also just measure Mahalanobis distance, or you can use a support vector machine. In any case, you can train a model to learn the statistical association between a pattern of voxel activity and a given stimulus. And this is what Daniel, I guess, was after there. So he showed different kinds of stimuli, and then likely he will be interested in looking at the differences in the pattern. What you do with this, or how it was framed for a long time, was kind of you decode what people saw from their brain. This actually also got me hooked on fMRI then around 2005, because I thought, well, that's a little bit better than this. If you really study this, you see that this and this measures statistical associations between the variations in the bold signal and the task. So it's not more awesome or better in any way. In general, it's actually from my statistical perspective, it's actually worse, because here you usually don't have a proper probabilistic model, so you end up with classification accuracies, and you don't have a really quantitative way to describe your scientific uncertainty. Doesn't stop people from hyping this, especially if you, for a classifier, use a neural network, which, as I said, was really not on vogue when I started fMRI. So people, when I talked about them at that point, and they actually used a neural network, they were like, well, neural network, you have to use support vector machine. And then in 2014 or so, it was like, deep learning, you need to use neural networks. I'm getting old, as you might figure, but yeah, it's around. It's all based on this. Who has seen a classifier paper or a representation similarity paper? So the other people don't remember. It's a little bit close to vision. So vision papers use this more often maybe than more cognitive-oriented paper. But then if you have a more cognitive-oriented paper and it uses a classifier, then it's awesome, because it uses a classifier. So this is from 2005 on. It is really a big that was really a big hype, and I also jumped in on this hype. So I have these very old papers from 2007 and 2008 using these approaches. But then I thought, well, that's fine, but one can do a little bit more. Maybe look at the differences in the patterns and how these are actually located in high-dimensional spaces. So this then became popular under the name of representation similarity analysis. But what I really wanted to do is basically, in this case, you also commonly average over time, and you don't have a notion of time, and everything in cognition evolves over time, right? So the typical thing in cognition is something happens, you process it, and you react. Yeah. So time is a crucial thing in cognition, and this also does not really appreciate this. So what I then became much more interested than using a different classifier algorithm is to actually look into time series models, so models that have an explicit notion of time, and later then also of space. And these, unfortunately, in cognitive neuroimaging come under the mysterious term dynamic causal models, which are not causal they're causal in the sense of time, so the four other things might be more causal than the other way around. But they are fairly mysterious for two reasons. First of all, because they rest on a differential equation framework, which most people in neuroimaging are not familiar with. And on top of it, they are inferred and fitted on using a variational based, which is actually quite an advanced machine learning or probabilistic modeling approach. Hence, you have a toolbox that does this DCM. In typical papers, you will see these kind of areas and then some parameters, but it will not really be clear to you what's actually going on. As I said, we will discuss this in some detail in the summer term. These, however, are also not the end of the story, definitely not from my perspective, because these are biophysical models. So with these you can nicely describe biophysical properties of the signal. You can build in biological assumptions like the hemodynamic coupling, neural activity, different neural populations, and so on. You can do this with these, but they then don't do anything. So let's say what they do is they do the signal, but they don't solve any tasks or problems. So really the future is the combination of this cognitive computational modeling with these biologically inspired time series models. So this is what I do want to do, have done, try my best. But the thing is, in the end, everything is always based on this kind of data, and all of these things are just ways to look at this data in different ways. Okay, I should stop this at this point. Questions?
Can I ask a question to the biological I'm just curious. I remember basically it measures the level of this deoxygenated hemoglobin and like Bolt signal. Yeah, I'm talking about that right now. But I remember in our brain there is this hematoencephalic barrier with astrocyte, and there is a brain liquid there. Like how it works with each other, doesn't it?
We will actually also talk about that. But the thing is, so there you of course have also very, very small vessels, right? So of course there is the blood-brain barrier, but and you don't measure the hemoglobin now somehow in a neuron, but you measure, of course, the hemoglobin where it's supposed to be. So the blood is still in a vessel, but it's in a very small kind of vessel.
So it's more in a big.
Yeah, that's also what we will come to that, but it's of course a very important point already, is that you don't get to a neural level resolution with the whole thing for two reasons. First of all, the physical system that you use for that, so MR machine, only allows for a certain level of resolution. but this can actually be pushed quite a lot, so you can actually get to micrometer resolution with MR. The other problem is, of course, that you don't have this one-to-one association between a single neuron and a single hemoglobin molecule that you can allocate there, but you have rather larger areas that become active, that draw in fresh blood, essentially, and if they do, then kind of a larger area, for a larger area, the signal goes up. So you definitely have a limit to the spatial resolution for fMRI from both physical constraints and biological constraints. Yeah. But we will actually talk about this as one part of this course. Anything else? No. Okay, then let's have a final break for today, and